Hi everyone, Dr. Becker. We are going to be jumping into chapter 5. This lecture is divided into two different PowerPoints. We'll start off with part 1. This chapter is going to discuss infectious disease. It's exciting that we're finally able to move out of these cellular type chapters and move into more disease states. To preface this, as we move into these type of chapters, remember, we would never have enough time to go through every associated pathophysiologic condition that's related to, say, infectious disease. You could take a course that's a year long between 2 semesters to cover infectious disease in more detail, which would be awesome. We don't have that capacity. So remember, this book goes through and again has to cover a lot of information in a small period of time. So we do move from topic to topic. Within each one, you're going to learn a few different examples of pathologic states, but certainly we're not able to learn all of them. We're going to cover the concept of a host, a pathogen, those type of things, how the human and the microbe organism, how we relate to each other, what's disease states, what's not, and how does that work within the body kind of concepts. The book starts with the top 10 causes of death worldwide. And this is from the World Health Organization. Please don't get caught up in specifics if you are an infectious disease guru. This is a contentious topic and people will argue the data, but for the purpose of us, we're working off of TAC's book and we're rolling with it. So on the right, you have the high income countries, and anything with an asterisk is infectious disease. For us, we have 5 diseases that are higher than any infection. So in America, the top 10 causes of death, and we're talking across America. So age, sex, color, gender, all the things, age, doesn't matter, the top 10. So we in America tend to die from heart disease, stroke, Alzheimer's, lung cancer, COPD. Then we get to lower respiratory infections, which typically are things like pneumonias. And then we have rectal cancer, colon cancer, diabetes, kidney disease, and breast cancer. Now, certainly if you were to start adding some filters to this and look at different cohorts of population, then we would have some different things, right? So if we looked at people over the age of 65 in America, what are they dying from? But again, this is just the top 10 causes of death across all ages, all factors. It is a contrast to the low-income countries because, again, not many people in America are truly going to die of diarrheal disease. That's the difference between low-income and high-income countries. So where we're going to focus on our attention is not to memorize necessarily malaria as much as we would want to know more about the common pneumonia. The interaction between the human and the microbe has a lot of factors that we have to understand. We have areas of our body that should be completely sterile, right? We know our blood, for example, should not have any bacteria or microorganisms in it. The other big one is our cerebral spinal fluid. The CSF should be a very protected liquid that should be clear, etc. You should not have any microorganisms in your CSF. The heart is another one. They really shouldn't have any flora, quote unquote, that are living there. But we do have normal flora or groups of microbes on other surfaces, and these are typically the ones that are exposed to the air. So if you think about our mouth and our skin, and then also things that we put into our body, so our GI tract and our urinary tract, and then for women, the vaginal area. Are common spots that if you were to swab it for bacteria or a microbe, you're probably going to have it be positive. That is what we refer to as colonization, because colonization is the presence of microbes on a body surface that does not cause disease to the host. So we call that like the normal flora. All of us, if we swabbed our skin, would probably come back with a staph, Staphylococcal aureus of some kind. But if I'm in good health, I'm colonized with that. That's where microbes are living, but they are not causing disease. Really important thing to remember is that at birth, right away when we are born, we are exposed to flora. It starts at birth. Some of the microbes are commensals that help maintain homeostasis while providing for their own needs for survival and growth. Some are not. So at birth, we are then exposed to the postnatal microbe exposures. and that can lead into colonization or versus an infection. It is really important to also remember that the human microbe interaction and for us to have normal flora is very, very important. Why is that? Because we need them. We actually have a symbiotic relationship with the natural flora that live. We know this, right, when we think about C. diff. We know from being in practice in the hospital, when we give a bunch of antibiotics and it alters the flora of our gut, that is then when it's a bad thing and we get bad bacteria override the gut and we get C. diff, right? We'll talk about that later. That's a perfect example. So we do have a good relationship, quote unquote, with that we need to remember is very helpful. The normal flora will help us with things like vitamin synthesis, our digestion function, right? We'll talk about that more in chapter 13. And then our ability to develop an immune system also requires this relationship. Skin is our biggest organ of our body, right? And so this relationship between skin and flora is huge. Our skin and our mucosal surfaces in our nose and our mouth, for example, are protected because they should have an intact barrier. Our structure should be intact. Also, we have secretions such as sweat and tears that contain antimicrobial compounds such as dermacidin and lysosome. So they have a function within our body that will affect that microbe relationship. This is that section from your book here that I'm highlighting and speaking about. So that's an important concept to remember, that our relationship between our human body and our flora that is naturally found is really important. Our innate and adaptive immune systems will provide the mechanisms to kill the invading pathogens and help us develop a specific immunity against recurring infections from such things. The book gives us this slide here that kind of shows some of the ones we're going to talk, it does show the ones we're going to talk about. So you can have this to help take notes as well. Understanding the difference between colonization versus infections listed here. If we are colonized with the flora, it's normal, we have no inflammation, it's limited growth, there's no signs or symptoms of disease, and we can call it a carrier state. When it moves into an infection, that means now that the pathogen numbers have increased and it's invaded tissues. We see the local inflammation, redness, heat, swelling, pain. If we're in a respiratory system, we have cough and airway irritation and mucus production. This can cause us to become septic or have bacteremia. So what are the things that affect it becoming a pathogen? Okay. So a pathogen requires a portal of entry. We have to have a way for it to enter to cause disease. So if you think about our skin, we have normal bacteria on our skin, and then we cut ourselves, and that bacteria in the area is allowed to invade, and then it becomes infected, it grows in numbers, we get an abscess, etc. So those are the kind of things we're talking about. We can inhale it, we can eat or drink it, we can come into contact it with microbes through the genital, urethral, or anal exposures. We can come in contact with the membranes of our eyes, nose, or ears. We can be bit, and the animal or insect can be the vector. We can have a penetrating wound. We can have contaminated blood. Remember things like transplants. Those are foreign things entering our body. And we can also have a surgical incision or a procedure done. Anything that's going to alter our human microbe interaction. The movement of the pathogenic organism within the body will depend on the structure of the pathogen and its capacity for being able to bind to human cells and tissues. So some pathogens are very invasive and they like human tissue and are very fast and they'll gobble right up, and others are really slow and harder to actually attach and cause disease. We'll talk more about that. The example your book gives is the hemagglutinin, which is a spike-shaped protein on the surface of the influenza virus. It attaches to host cell celiac acid molecules on the membrane of our respiratory tract cells, and then those targeting those cells for invasion and replication, right? Think about some things such as an artificial heart valve or an artificial joint. That would also provide an optimal environment for bacteria to form a biofilm that is highly resistant to immune attack. We see this all the time in the hospital. If someone gets a joint infection and they have metal in that joint, oftentimes if we pull a culture from the area and it shows a specific pathogen, they're going to have that joint removed and take the hardware out, because we know a biofilm will adhere to that metal surface and nothing we're going to do is going to get rid of that bacteria. So how the pathogen is, how virulent it's going, or virulent, however you say that, I say virulent, is going to be within our body depends on a wider range of factors. We know that the extreme ages, so extremely young, extremely old people are more at risk because their immune systems either aren't developed or they're weakened. If we're malnourished, if we're in bad health, if we have a bad disease state like uncontrolled diabetes, all of those factors would come into play as to why one person would get sick from a pathogen versus another. When we talk about some of the pathogenicity of some of these diseases, we have to discuss these things that are listed on the slide here. A lot of what we're talking about is first going to become revolve around bacteria. So when we're talking about these, a lot of this is true to bacteria. There could be some other nuances if we were talking about, for example, a virus. So first thing is mobility of the pathogen. So that's its ability to adhere to membranes and to migrate along them or move to other tissue layers and organs. If it's highly mobile, it's going to be more virulent than if it has low mobility or poor mobility. Then we talk about the ability to resist breakdown by lysosomal enzymes. Okay. The ability to adapt to new environments is important. H. pylori can thrive in acid in our stomach because it thrives there. So where do we look for H. pylori? Mainly in the stomach. That's where it's going to be most commonly found because that's where it can survive. A pathogen's ability to evade our natural immune system to be detected or destructed, it's going to be a very bad pathogen. So the example they give here is the Staph aureus can live inside of the neutrophils and then invade lysosomal destruction. We'll talk more about that. The pathogen's secretion of exotoxins that destroy host cells and organ functions. The pathogen can cause rupture and release of endotoxins that provide hyperactive immune reactions. Then we can have release of superantigens that further provoke immune reactions. Some of the other things that are at play here is the secretion of the exotoxins that can spread via the bloodstream and disrupt tissue function in other places, and the ability to resist or counter the effects of the pharmacological agents given. So all of those things are going to play a part in how pathogenic a microbe or bacteria or virus is. So our host, as us as the human body, right? This is where I said our age and our general level of health obviously are at play. Older adults can be very vulnerable because they may already have a diminished immune response. That would make the onset of infection more subtle and the diagnosis more difficult. Very, very true in adult gerontologic medicine. Not all geriatric patients are going to spike a fever of 103. Not all of them are going to get a white count of 30,000. They can have a weakened immune response, and so fever and white count can be subtle abnormalities that can really make diagnosis more challenging. Anybody who has an uncontrolled disease state, okay, or have other factors going on that are going to affect our immune system. So if you are on immunologic therapy, you're on Humira, you're on those other biologics that are suppressing your natural immune system, you are certainly going to be more at risk for infection than somebody else. Think about people that have cancer going on. Their immune system's affected, and they're going to be more prone to infection and to get sicker than people who have an intact immune system. They talk about the Spanish flu epidemic of 1918. It had high mortality rates in healthy young adults between the ages of 20 and 40, as well as the young and the old, right? So the magnitude of the host immune response can also amplify the severity of the disease progression. So in the Spanish flu, the apparent mechanism for that pattern was the robust inflammatory response young adults had. Their host response to the flu virus was so intense that the massive cytokine release resulted in septic shock. So remember that healthy people can get very sick very quickly as well. I have cared for healthy athletes, 20-year-old males that have died from influenza because they can have a very robust response, which actually predisposes them to alveolar flooding and actually caused their respiratory failure to be way more extreme than an 80-year-old, and they had significant compromised gas exchange. Most of the organisms on this planet are not pathogenic. The microorganisms with the capacity to cause disease to humans are not always going to be pathogenic. It's estimated that there's about 1,400 pathogenic microorganisms, which is a minuscule fraction of the estimated 1 trillion microbial species on Earth, right? So again, not every microbe that we find on the planet Earth is going to cause infection. Most of the microorganisms that live and thrive on humans, which is normal flora, and the text calls them the commensals, normally do not cause disease without some precipitating or inciting event. However, infectious diseases remain a global threat to public health. We have to remember that we're talking about bacteria, fungi, protozoas, parasitic worms. We also have things that are non-living viruses and prion particles. Many of the organisms that are present in our environment can also interact with other non-human hosts and also affect us as well. We have relatively few microscopic pathogens that have humans as our exclusive hosts, meaning that those organisms can only be transferred from human to human, right? A lot of our bacteria we also share with our pets, our cows, our animals, or dogs, or other insects or animals within our environment. We usually typically will label the transmission of an infectious disease as either direct or indirect. So direct would be like person-to-person contact, whereas indirect would be like airborne spread, animal bite, touching a surface and you get a contaminated surface with E. coli or salmonella, those kind of things. All right, going to the different types of pathogens and their classes. So we start off with bacteria. It seems like when we're talking about infectious disease, the majority of the time we're talking about bacteria. But as you'll learn in your acute care path, we also have lots of other things that can be pathogenic. But let's start with the bacteria. Bacteria are prokaryocytes, so they're single-celled organisms that are much smaller than human cells that reproduce by binary fission. The different species of bacteria have varying shapes and sizes, right? So we call it diplococci, those kind of things. They form simple spheres and rods or spiral shapes and other such configurations, right? So this is diplococci, streptobacillus, all of these different things. So the cocci are spheres found as unitary bacteria or in typical clusters such as diplococci, staphylococci, or streptococci. Bacilli are these rod shapes And then there's some more complex shapes, which include the helical and budding forms. We're going to jump here a moment, though, and go back up here where I've added our link to our osmosis, and let's listen to what Elsevier Osmosis has to say with some great illustrations.
Bacteria are prokaryotic cells that play an important role. They can cause disease, but are also part of the human microbiota and live on our skin, body, and on everyday objects in our environment. When compared to eukaryotic cells, the structure of bacteria is less complex due to a lack of nucleus and membrane-bound organelles such as mitochondria, endoplasmic reticulum, and Golgi complexes. Now, bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. Let's start with the cell wall, which is found on the outer surface of the cell membrane, and its most important role is to protect the bacteria. It consists of peptidoglycan, which usually helps differentiate whether bacteria are gram-positive or gram-negative. Gram-positive bacteria have a single thick layer of peptidoglycan above the plasma membrane, which allows them to retain the staining dye. And gram-negative bacteria have a thinner layer of peptidoglycan sandwiched between the surface membrane and the plasma membrane, so they can't retain the dye.
Sorry, I jumped in and hit the wrong button. I do want to point out here that this is really important content to understand the cell wall of bacteria, to understand how we can send something to the lab and find out if it's gram-positive or gram-negative. These are really important concepts to understand because when you move forward into your career and you're practicing wherever you are, if you have anything to do with writing antibiotics or antiviral medications, this is really important content to understand because you're going to hear the things, you have a diplococci gram-positive, da-da-da-da-da. And to really understand what that means is very important. So keep going.
So they can't retain the dye. Additionally, the cell wall helps maintain their shape. The round-shaped bacteria are called cocci. The rod-shaped ones are called bacilli. Spiral-shaped ones are spirilla. And sometimes the same bacteria can have multiple forms, in which case they're called pleomorphic. Some bacteria are covered by a capsule, which acts as a shield that protects the bacteria against phagocytosis and also helps the bacteria adhere to surfaces. The capsule is considered an important virulence factor since the strains that lack a capsule are less virulent. Underneath the bacterial cell wall, there's the plasma membrane, which is the most important layer because it encloses the cytoplasm, which is a gel-like substance composed mainly of water that also contains cell components, enzymes, and various organic molecules. If the plasma membrane is removed, the cell's contents spill into the environment, and the cell no longer exists. Now, the plasma membrane is responsible for most of the cell's relationship with the outside world, by acquiring nutrients and eliminating waste, and also maintains the interior of the bacteria in a constant, highly organized state. Usually, all plasma membranes are selectively permeable barriers, which allows certain ions and molecules to pass in and out of the cell while preventing the movement of others. However, in bacteria, the plasma membrane has other important roles, such as respiration, photosynthesis, and the synthesis of lipids and cell wall components. Now, in the bacterial cytoplasm, there are scattered cell components such as ribosomes, which are the sites of protein synthesis. These ribosomes can be found spread all over the cytoplasm, where they produce proteins that are destined to remain inside the cell, or they can be attached to the plasma membrane, and they are called plasma membrane-associated ribosomes. And in this case, they make proteins that will reside in the cell envelope or get transported outside the cell. Another structure found in the cytoplasm is the nucleoid, which is an irregularly shaped region that contains the cell's chromosome. The chromosomes of most bacteria are a single circle of double-stranded DNA, but some bacteria can have a linear chromosome, while others can have more than one chromosome, and these chromosomes contain the genes needed to provide instructions for protein synthesis in the cell. Additionally to the genetic material contained within the nucleoid, many bacteria contain extrachromosomal DNA molecules called plasmids. These plasmids are small, double-stranded DNA molecules that can exist independently of the chromosome, and they can be circular or linear. Plasmids have relatively few genes, and their genetic information is not essential to the bacterium, and cells that lack them usually function normally. However, plasmids often contain genes that confer a selective advantage to the bacterium, such as resistance to antibiotics. Finally, the The bacteria can move or attach to other cells and surfaces with the help of structures that can be found on their external surface. So they have pili, which are hair-like structures that allow them to attach to other cells, and when these are shorter, they're called fimbriae and help the bacteria attach to surfaces. They can also have flagella, which are long, whip-like protrusions that allow the bacteria to move. Alright, as a quick recap, the bacteria are prokaryotic cells that play an important role in human disease and health, and can be found almost anywhere in the environment. Bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. They have a cell wall which protects the bacteria, determines whether they are gram-positive or gram-negative bacteria, and also helps maintain their shape. Some bacteria can also have another extra layer on top of the cell wall called a capsule, which is a major virulence factor. Underneath the cell wall, there's the plasma membrane, which is the most important layer because it encloses the cytoplasm and defines the cell, having important roles such as respiration, photosynthesis, and the synthesis of lipids and cell wall constituents. The cytoplasm contains ribosomes, which are the site of protein synthesis, the nucleoid, which contains the genetic material, and plasmids, which contain genes that confer a selective advantage to the bacterium.
All right, at the end of this video, we have a quiz summary. So let's go ahead and take this to review this content here and see how we do. So a researcher is studying a bacterial strain. The bacteria are found to produce a thick-walled intracellular structure when exposed to harsh environmental conditions. This structure enables the bacteria to become dormant, thereby minimizing metabolism. Which of the following pathogens is this researcher most likely studying? So I bring this up because this is a perfect example of questions that you could have further down the line for different things, for tests, for pathophysiology, for the different things that move on. So let's do our elimination tool. Elimination tool, what is that? Oh, sorry, I'm thinking of the wrong. So let's see, which one, a thick-walled cellular structure. What becomes dormant pretty easy, and then comes back up to bite us in the tushy. So let's see. Actually, sorry, I changed the screen. That was weird. Okay, so we were talking about this one. My guess is going to be the Clostridium botulinum. I feel pretty good about it. All right, so just helpful, I think, to understand some of the concepts around what we're talking about. So back to the text here. They're small prokaryocytes. There's many different shapes. They reproduce by binary fission, not mitosis. There's no nucleus. So the DNA makes up the nucleoid and it's coiled in cytoplasm. The cell wall is external to the cell membrane. Here we are talking about the properties of the Gram stain, which the osmosis video illustrated well for us. Gram-positive bacteria have the thick cell walls rich in peptidoglycan that can retain the stain. Gram-negative have a thinner cell wall that do not retain the stain after the procedure. Instead, you have to have a counter stain that will make them visible. There are some bacteria, such as the mycobacterium, some bacteria, such as the mycobacterium, that do not respond reliably to the Gram stain, so they will have different tests and different nomenclature. Remember that I'm going to add that in here, that it's important to remember that we can be aerobic or anaerobes. Can I spell anaerobes? There we go. Anaerobic. So anaerobes thrive in high oxygen areas, and anaerobes thrive in low oxygen areas, and some bacteria can actually be both. Okay. Remember that bacteria otherwise lack intracellular organelles. And then they talked about in the text too, the projections assist adhesion and mobility and the different tissue invasion, such as the pilus and the flagellae. Okay. So the example your book gives is that gonorrhea membranes are studded with projecting pili that allow the adherence to the genital mucosa and movement along the epithelial cells to spread the genital infection. So the severity of infection is influenced by, remember, our virulence factors that can include the structural elements of the bacteria, its ability to enter the cells, and the capacity to produce damaging secretions such as degradative enzymes and exotoxins. The other example that the book talks about is the Mycobacterium, so TB, avoids further destruction by means of its thick, lipid-rich cell wall and its ability to block lysosomal degradation. Certain bacteria are capable of forming spores, which are dormant structures with impermeable capsules that can exist without nutrition and evade destruction for extended periods of time, then only to be reactivated to their original form when certain conditions permit it. Okay. The other kind of components of bacteria that we need to talk about is the bacterial proliferation and activation that will lead to an infection. So our host inflammatory and our immune response is what contributes to the characteristic systemic signs of infection. So we're thinking fever, malaise, redness, swelling, pain. But also remember, osmosis talked about exotoxin secretion. So these exotoxins may have pathophysiologic effects on the target cells via direct damage or interference with host cell function. Two such examples will include cholera toxin, which promotes massive watery diarrhea, and botulinum toxin, which causes neuromuscular dysfunction and paralysis. Endotoxin release. Endotoxins are lipopolysaccharide molecules that present in the gram-negative bacterial cell wall. As antibiotics or the host complement system will work to defend the host, they may rupture the bacterial cell wall, and that releases these toxic fragments that can provoke a systemic inflammatory response and then can cause septic shock. I have added a Microsoft Word document for you to please review. It's just got some general information on exotoxins and endotoxins with some more information for you to know. I wanted to add information for you as I don't feel like the book goes into it quite as much as I would like for you to know. So please review this material on the Word document and know that some of this information on here could possibly be testable material. This is important things to understand about the endotoxin and endotoxin and what their difference is. Okay, I'm not going to get crazy where I want you to memorize all of these things, but know where this document is and have it ready if you have questions about endotoxins or exotoxins moving forward. Next, we're going to talk about the features of the bacteria structure. This was also talked about in osmosis. This is an image from your TCAX or your TAX book. A bacterium has the multiple surface layers, an inner plasma membrane, a cell wall, and in some species it does have that protective capsule. The complexity of the cell wall differentiates gram-positive and gram-negative organisms. Remember, we've talked about this quite a bit, and that's Gram positives keep the stain, gram negatives don't keep the gram stain, so they need to have their other microscopy procedures. Remember that the flagella allow movement within the host, and the pili facilitate attachment to the host membranes. DNA is found in that large circular chromosome, which we call the nucleoid, and it's located within the cytoplasm, which is different. It's not bounded by the nuclear membrane as in eukaryotic cells. So the difference with the bacterial cell is that the DNA is found in the nucleoid within the cytoplasm. Additional DNA may be found in smaller circular plasmids, which aren't shown here. When bacteria is reproduced, it's by binary fission, which is different than other cells. The DNA in that nucleoid and those plasmids are replicated and then divided among daughter cells. This method of reproduction is faster than mitosis, which is bad because, as we know, bacteria can replicate very fast, which is why they can be very dangerous. The ribosomes are used for protein translation, which is similar to the eukaryotic cells. The differences between prokaryotic and eukaryotic cells provides the foundation for drugs used to treat bacteria infections. So, clinical aspects of bacterial infections on here. The bacteria can duplicate rapidly by binary fission, which we've said. The doubling rates or the generation time allow for exponential growth soon after an infection. Some bacteria can divide every 20 minutes. Growth projections in the body during infection are not known and could be slower than what we've seen in the lab, but still we know that people can almost have a fire spread of bacteria. In our bodies, the presence of the bacteria stimulate a host response by macrophages, mast cells, and neutrophils. The release of the cytokines, particularly interleukin-1 and tumor necrosis factor alpha, activate our immune response and cause signs and symptoms like the fever, malaise, and loss of appetite. We're going to talk more about this in our next chapter. Some bacteria release the exotoxins that act on the target organs, right? Cells of gram-negative bacteria release our endotoxins when lysed. So endotoxins further stimulate the white blood cell cytokine secretion, and they worsen the sickness-associated signs and symptoms. So when endotoxins are present, people tend to get a lot sicker a lot faster, and they're a lot more dangerous. So briefly here, the book also does a quick job here of showing you this figure 5.4. So on this side in A here, we're showing the earliest these are the antibacterial drugs that are targeting the differences between the prokaryotic and eukaryotic cells. And they work by disrupting cell wall synthesis, nucleic acid synthesis, and protein synthesis. So this side here, that's 1. This shows the earliest widely used antibiotics. They would target the synthesis of the cell wall components, and new beta-lactam drugs, which are penicillin, cephalosporins, carbapenems, monobactams, are continuing to be developed. Early drugs, like our early true penicillin, were most effective at eliminating gram-positive bacteria due to their thick peptidoglycan layer, whereas later generations of drugs gained by the gram-negative bacteria coverage. So the penicillins and these other early microbials contained the beta-lactam ring, which here disrupted this cell wall and allowed it to work and kill off the bacteria. The nucleic acid synthesis, so remember I'm giving an antimicrobial, so an antibiotic, that is now trying to disrupt nucleic acid synthesis over here. So on a bacteria cell, the nucleic acid synthesis was found to be disrupted by another class of antibiotics, which were the sulfonamides. Please pay attention to this. This is going to be very helpful information when you also move on to pharmacology. So sulfonamides, we're thinking like Bactrim. They block the sequential steps in the conversion of the precursor, the PABA, the para-aminobenzoic acid. So they block the conversion of PABA to DHFA, which is dihydrofolic acid, and to tetrahydrofolic acid, THFA. So again, the sulfonamides and trimethoprim will block the sequential steps here, and these will then block the conversion of PABA to DHFA and to THFA. And those are the steps that are required for the purine synthesis needed for DNA replication. Okay, so if you were to ask the question, what do beta-lactams do? What's their mechanism of action to work? We would say that they disrupt cell wall synthesis because they disrupt the cell wall formation. If someone were to ask you, how does Bactrim work? What's sulfonamide and the trimethoprim? What's its mechanism of action? We would say it disrupts nucleic acid synthesis because it prevents the conversion of PABA to DHFA to THFA, which is required for purine synthesis and DNA replication. Okay. If someone were to ask you, how do the quinolones work, like levofloxacin, Levaquin, who's a fluoroquinolone, what's their mechanism of action? We would want to say it interrupts the DNA gyrase and the RNA polymerase here, which is also disrupting nucleic acid synthesis. Then we can go down here, and these over here would be drugs that are going to block the protein synthesis. Some antimicrobials block bacterial ribosomal subunits. Here, 50S and 30S. These terms are going to come back up when we get to pharmacology. So some of these drugs that will block bacterial protein synthesis would be macrolides, so like azithromycin, clindamycin, linezolid, tetracyclines, aminoglycosides. Some of them specifically target the bacterial ribosomes while having no effect on eukaryotic ribosomes. Then over here, these other ones like vancomycin and bacitracin also will disrupt the cell wall in a different way. And then like the polymyxins, erythro, things like that also disrupt the cell membrane, but we're talking about those in depth. Before we wrap up the end of the bacteria, I meant to put this up there, and I'll add the link to this in your readings. I wanted to, again, just talk quickly about the difference between the prokaryotic and the eukaryotic cells, okay? Because I think these terms start to get a little confusing. So this is also from osmosis. So eukaryotic cell is a cell that contains a membrane-bound structure, and it's the basis for every multicellular organism, including animals, plants, and humans, right? And such things like a protozoa. But a eukaryotic cell contains several membrane-bound structures, or those organelles, which are specialized cellular subunits that carry out specific cellular function. The nucleus is surrounded by the nuclear membrane, also called the nuclear envelope, and this protects the genetic material stored inside. The nuclear membrane has nuclear pores, which are selectively allowing only certain substances to pass through. Another membrane-bound organelle is the endoplasmic reticulum. Remember, we have rough and smooth. The rough ER extends from the nuclear membrane, and it's covered with ribosomes, and that's the location of protein synthesis. Meanwhile, the smooth endoplasmic reticulum is the main site of lipid and steroid synthesis. Each eukaryotic cell can specialize and contain different proportions of each organelle type depending on its function. Gives us some ones there. So what's the difference between the prokaryocyte and the eukaryocyte? The primary difference is that a nucleus and other membrane-bound organelles are only present in eukaryotic cells. Prokaryotes are always unicellular, while eukaryotes are often multicellular. Okay. So this kind of talks about that. So some examples of the eukaryotic cells are plants and animal cells. Okay. What's the most important facts to know is, again, the eukaryotic cells contain the membrane-bound organelles and are the basis for both unicellular and multicellular organisms. Prokaryotic cells do not have any membrane-bound organelles and are always part of a unicellular organism. So when you think about it, the prokaryocytes are the single-celled microorganisms that were found the earliest on Earth. And typically, when we think prokaryocytic cells, we're thinking bacteria, etc. Okay? So I just wanted to give that kind of brief overview before we jump out of bacteria and into viruses. So next, your book jumps right into the viruses. I'm going to jump over to osmosis here, and let's just watch that real quick. Osmosis does a great job, again, of giving us the illustrations, audio-visual, on the review of viruses and their structure and functions. Let's jump in here.
And eventually released.
Let's start in the beginning. That'd be good.
Viruses are a unique group of pathogens with a simple acellular organization and a distinct pattern of multiplication. Despite their simple structure, they are a major cause of disease. They have no cytoplasmic membrane, cytosol, or functional organelles, but they can infect all types of cells, and numerous viruses can also infect bacteria, which are called bacteriophages. Viruses and bacteriophages are not capable of metabolic activity on their own, so instead, they invade other cells and use their metabolic machinery to produce more viral molecules, nucleic acid, and proteins, which then assemble into new viruses. Viruses can exist either extracellularly or intracellularly. In the extracellular state, the virus is called a virion and isn't capable of reproducing. A virion consists of a protein coat called a capsid, surrounding a nucleic acid core, which contains the genetic material, or the viral genome. The nucleic acid and the capsid are collectively called a nucleocapsid. Some virions have a phospholipid membrane derived from the host cell called an envelope, which surrounds the nucleocapsid. The viruses that have an envelope are called enveloped viruses, and these include the herpes viruses and HIV, while the ones that lack the envelope, such as poliovirus, are called non-enveloped or naked viruses. Once inside the cell, the virus enters the intracellular state, where the capsid is removed and the virus becomes active. In this state, the virus exists solely as nucleic acids that induce the host to synthesize viral components from which virions are assembled and eventually released. Now, the viruses are surrounded by an outer protein coating called a capsid, which protects the viral genome and aids in its transfer between host cells. Also, according to their capsid symmetry, the viruses can come in many shapes and sizes. There are 3 types of shapes: helical, icosahedral, and complex. First, the helical viruses have a capsid with a central cavity or a hollow tube, which is made by proteins arranged in a circular fashion, creating a disc-like shape. The disc shapes are attached helically, creating a tube with room for the nucleic acid in the middle. An example of a virus with helical symmetry is the tobacco mosaic virus, which is a most studied example. Moving on to the icosahedral viruses, which are made up of equilateral triangles fused together in a spherical shape that fully encloses the genetic material. These viruses are released into the environment when the cell dies, breaks down, and lyses, thus releasing the virions. Some examples of icosahedral viruses include poliovirus, rhinovirus, and adenovirus. Finally, the complex viruses have a combination of icosahedral and helical shape, and may have a complex outer wall or head-tail morphology. The head-tail morphology is unique to viruses that infect bacteria, called bacteriophages, and they have an icosahedral-shaped head and a helical-shaped tail. These viruses use their tail to attach to the bacterium, and then they create a hole in the cell wall and insert their DNA into the cell using the tail as a channel. Now, the nucleic acid core of the virus contains the genetic material, which can either be DNA or RNA, and the total genetic content of the virus is called the viral genome. The viral genomes are usually small and contain only a few genes that encode for proteins needed by the virus that's not present in the host cell. The genetic material can be single-stranded or double-stranded, linear or circular. In DNA viruses, the viral DNA enters the nucleus and uses the host's DNA polymerase to create more copies of itself, as well as viral mRNA that gets translated into viral protein. With RNA viruses, many of them stay in the cytoplasm, where the viral RNA is used by the host's ribosomes to create viral RNA polymerase proteins. These will help replicate the viral genome, so more viruses can be created. RNA polymerases are more likely to make copying errors than DNA polymerases, and therefore often make mistakes during transcription. This is why mutations in RNA viruses occur more frequently than in DNA viruses. Alright, as a quick recap, viruses are a unique group of infectious agents with a simple acellular organization and a distinct pattern of replication. They can exist either extracellularly, where the virus is inactive and isn't capable of reproducing, or intracellularly, where the capsid is removed and the virus becomes active. A virus in the extracellular state is called a virion, and consists of a protein coat called a capsid, which protects the viral genome and aids in its transfer between host cells. The capsid is surrounding a nucleic acid core, which contains the genetic material, or the viral genome, which can be either DNA or RNA. Some virions have a phospholipid membrane derived from the host cell called an envelope, which surrounds the nucleocapsid. According to their capsid symmetry, the viruses can come in many shapes and sizes, and they can have 3 types of shapes: helical, icosahedral, and complex.
Great review from Osmosis, reviewing the viruses, their structure and function. Let's talk through a couple points from your text to make sure we highlight these. The viruses are acellular, which they talked about. They must enter the host and they use the host, okay? So the virus uses the host enzymes and ribosomes to conduct the nucleic acid replication, which we saw there. So they have to use the host enzymes and ribosomes to do the nucleic acid replication, transcription, and translation in order to reproduce themselves, and that requires using the energy of the host cells. They talked about the fact that a mature extracellular form of a virus is called a virion. All virions have similar components, that they have the nucleocapsid composed of the DNA, RNA, surrounded by the protein capsid. And viral nucleic acids can be RNA or DNA. They did a great job illustrating that for us. But remember that only one will be present. They're not like the other prokaryotic or eukaryotic cells. In viruses, they only have RNA or DNA, and so they could be either single-stranded or double-stranded. Remember those shapes that they talked about on there? I like that they gave us images of what those look like. The enveloped viruses have an additional outer membranous envelope surrounding the nucleocapsid that is usually derived from the host cell plasma during budding. Capsid proteins for those non-enveloped viruses and the spike proteins projecting from the membranes of the enveloped viruses are the antigenic determinants that provoke the host immune response. So the host immune response will form neutralizing antibodies and activate cytotoxic T cells then to kill the host virus infected cells, displaying the fragments of those proteins. So this here, excuse me, is the basic viral life cycle. And it begins with attachment to a host cell. We can open this up a little bit. So we have binding to the cell surface receptors, and then it has to enter the cell, right? We can see here it's bound, and then it enters, breaks through into the cell. So it removes the capsid coat, and then the viral nucleic acid is exposed. So it has this uncoating process here. And the viral polymerase, as well as the host cell polymerases and ribosomes, are then used, right? And they serve the functions that are listed in your book. Okay, so they will replicate many copies of the viral nucleic acid. Then they will transcribe the viral genome. So we have replication, transcription. Then they will translate the viral proteins. And then there will be assembling of the new virions, which are those mature cells, and hundreds can be produced at this stage here, right? So they've done all of those things utilizing the host cell energy, the host cell's energy. So they have to use our host enzymes and the ribosomes in order to do the replication, transcription, and translation in order to reproduce these multiple virions. And then the virions are locally or systemically released. Okay. So viruses have to kind of go through this process utilizing the host enzymes and ribosomes. There's the 1, 2, 3, 4, 5, 6, 7, 8 kind of the processes there that it works through. The book also talks a little bit about how do we have a portal of entry. I'm sorry, went to the wrong slide. So when we talk about the portal of entry here, and how does the virus come in then to bind to the cell surface receptors? So it's important to remember that there's a lot of ways that we can have the viruses enter us as a host. So we can have aerosols, so when we inhale droplets into the respiratory system. So let's think of the common cold, coronavirus, rhinovirus, adenovirus, the flu, and measles and mumps are actually inhaled into the respiratory system. So we can inhale it, we can digest it or ingest it with things like hepatitis A and E, norovirus, rotavirus. Remember those ones that cause us the horrible diarrhea. We can get a virus through sexual contact, HPV, herpes, HIV, hepatitis B. Obviously, we know that we can also get the viruses through blood or injection, HIV, hepatitis B and C. Also remember that fancy term we learned a long time ago in undergraduate of vertical transmission, meaning that you can have a mother to neonate transmission, which can be HIV and hepatitis B. This is important to discuss because in some cases, the viral replication involves the entry site. So where it comes in as an entry site can be important to understanding that virus. So especially things like the viruses that give us our upper respiratory infections. In other cases, though, the virus move from the entry site to their cellular target, and that happens like with hepatitis and HIV. The localized response produces cell death that can be observed on our slides in the lab, and that is referred to as the cytopathologic effect. So that's the response of the virus. Local stimulation of our immune system will cause those characteristic signs and symptoms that they're going to be seeing. So cough, cold, runny nose, those kind of things lets us know we're thinking the common cold. Other thing to remember is the time of infection varies. Remember that you can be exposed to the virus days before you get symptoms. A very virulent active virus. So it is very active, it replicates quickly, and it's highly contagious, typically, is what we refer to as the virulent viruses. Think of the influenza, for example. But other viruses may not be completely cleared, and they can enter that latent phase. And so when they're latent, no viral replication is happening, and they're not being spread. But then at later time, exposure to stress or your altered immunity allows resumption of the viral life cycle, and then the disease is expressed again. So we think of shingles, for example, right? We know that the herpes zoster. If we were exposed to herpes like varicella in childhood and then it went latent and dormant, and then we have a stressful time into adulthood, or we are put on strong immunotherapy or steroids, and all of a sudden it is allowed to be re-expressed. And this is also what happens with CMV, Epstein-Barr, and then like also your genital herpes. You can also have chronic and persistent viral infections, where after your acute initial infection, the virus replication drops to very low, and it's continued to be produced at such low levels, and it slowly has the capacity to cause slow accumulation of cell and tissue damage. So a person can be a carrier, meaning I'm able to affect others if I'm a carrier, without showing signs of the infection. So it's kind of this smoldering is the word I like to think of viral infection, like hepatitis C and like HIV. So kind of the key concepts here that we're thinking about when we're kind of wrapping up the life cycle of the virus. Each virus has that nucleic acid core, DNA or RNA. It is a more complex virus if it has a capsid protein coat. Also more complex if it has that outer membrane-like envelope. The virus has to attach to and enter into a host cell, and it uses the host cell's machinery to replicate its genome and synthesize capsid proteins. The viral particles are assembled and released from cells by budding. As intracellular pathogens, they are more difficult drug targets. So we believe that vaccines are very useful because we have good data to show they really help and they really work. Review one more time. We have to have attachment to the target cell by a cell surface receptor. And this is usually a highly specific recognition site on the outer surface for a very specific virus to bind. It has to enter the host cell by endocytosis. Then we have to have the removal, the uncoating of the capsid coat to expose that nucleic acid. Then we have to have replication of the DNA in the nucleus of the host cell, or the replication of the viral RNA in the cytoplasm of the host cell. So if you were to talk about the difference between DNA replication and RNA replication would be that noted there, that replication of the viral RNA is in the cytoplasm of the cell. And then you have to use the host cell's ribosomes to go through that transcription and translation of the protein. And then the virions will be assembled and then they're released, which may result in cell lysis. For those enveloped viruses, the virions acquire a membrane coat by budding off from the host cell. Okay, so when we talk about antiviral drugs, then how is that going to work? Well, obviously we want to disrupt this whole process, right? So let's talk more about that. Unfortunately, Osmosis does not have a video that I think is really good for what we're trying to do. So this is a gentleman called Speed Pharmacology. I think he does a pretty good job. This is a YouTube video, so let's watch this one and then we'll jump back into the text.
Antivirals are a large and diverse group of agents that are typically classified by the virus infections for which they are used. For simplicity, we can divide them into 4 groups, that is, number 1, anti-influenza drugs, number 2, anti-HIV drugs, number 3, anti-hepatitis drugs, and number 4, anti-herpes drugs. RNA. The viral
Sorry, hit a button again.
And that segmented RNA. The viral envelope is made up of a lipid bilayer that contains 3 of the viral transmembrane proteins, hemagglutinin, neuraminidase, and matrix 2, abbreviated as M2. The first step of the influenza virus infection involves binding of hemagglutinin to sialic acid residues on the surface of host cell, which results in engulfment of the virus into the cell via endocytosis. Next, the membrane protein M2 forms pH-gated channels that allow protons to move through the viral envelope and acidify the core of the virus, causing release of the viral RNAs and proteins, which are then transported into the cell nucleus. In the nucleus, viral RNA is transcribed into messenger RNA. The translation of mRNA into viral proteins takes place in ribosomes in the cytoplasm. Once synthesized, hemagglutinin and neuraminidase glycoproteins are secreted through the Golgi apparatus onto the cell surface, where they fuse with the plasma membrane, whereas other viral proteins migrate back to the nucleus, where they assemble with newly replicated genomes. Assembled viral capsid then moves to the plasma membrane, where it buds off, taking a segment of membrane containing the hemagglutinin and neuraminidase, and thus forming a viral particle. In the last step of the replication, viral neuraminidase enzyme cleaves and removes sialic acid receptors from the surface of the cell, thereby allowing release and spread of the virus to new cells. Now, there are three classes of approved drugs for influenza. The first one are M2 ion channel inhibitors, which work simply by blocking the M2 channel and thereby restricting passage of protons, which are required to trigger the release of viral genes into the host cell. Drugs that belong to this class include amantadine and rimantadine. Now, moving on to the next class, which actually represents a new class of anti-influenza agents called endonuclease inhibitors. At this time, the only agent in this class is a drug called baloxavir, which works by selectively inhibiting cap-dependent endonuclease, a key enzyme involved in the initiation of influenza virus mRNA synthesis. By blocking this enzyme, baloxavir prevents viral gene transcription and ultimately influenza virus replication. Finally, moving on to the third class of influenza drugs, that is neuraminidase inhibitors. The agents in this class exert their antiviral activity by inhibiting the viral neuraminidase enzyme found on the surface of the viral particle. Without active neuraminidase, the virus is unable to cleave sialic acid and unable to escape from the cell. Drugs that belong to this class include oseltamivir, peramivir, and zanamivir. Now let's move on to our next group of antivirals, that is anti-HIV drugs. So HIV or
Review of an antiviral drug for the flu, which is also what the book talks about in that section there. We are going to review next. We're going to stop with the viruses. The book moves right on into fungi and yeasts. Woohoo! Always a fun one to talk about. So the key elements here are listed. We think there's about almost 4 million species of fungi, and the vast majority are non-pathogenic, meaning they're completely benign. They're not going to cause us disease. But for several, such as Aspergillus and Cryptococcus, the human host is not the intended habitat and can actually be a challenging growth environment. Okay, fungi are eukaryotic cells. We talked about this. So they have multicellular things, and they are defined with defined nuclei and organelles. So they can be yeasts, molds, or both, and those are called dimorphs. Molds are multicellular organisms that reproduce sexually or asexually by branching hyphae, whereas yeasts are unicellular organisms. So one of your main difference between mold and yeast is molds are multicellular and yeasts are unicellular. Fungal toxins can cause disease in humans, and airborne fungi are a frequent cause of allergies and disease. Fungal infections in humans tend to be either superficial, so we get a lot of them on our skin, as we know, such as dermatophytes, like in our skin or our hair, or they can be systemic. Systemic is much less common. It's much more common to have a skin or a topical kind of fungal infection. But systemic fungal infections can be lethal. They're very hard to treat. Other examples of the superficial fungal infections are like Tinea pedis, which is fungal or athlete's foot. Then we have Tinea capitis, which is ringworm of the scalp. And then Tinea corporis, which is ringworm of the body. Another term, again, that we talked about here is that these are dermatophytes. And they're called that because of their association with the skin and their ability to get their nutrition from the keratin of the skin or our nails. We also get a lot of fungus on our nails, especially our toenails. Okay. This is a very important point here when we're talking about our practice, when we're going into the acute gerontological adult field. Fungal and yeast infections are much more common in an immunocompromised host. The book talks specifically about Candida albicans, which is a yeast often responsible for our oral infections such as thrush, and it's also one of the most common vaginal infections. Individuals who are immunocompromised, or also think of people who have artificial implants, prosthetic valves, or catheters, or people, of course, who have chronic disease. Those are the people that are more likely to get the systemic life-threatening fungal infections. When we think about the yeast, the common ones you're going to hear about are Candida albicans, Candida glabrata, and Candida parapsilosis. Those are the ones that are the most common invasive candidiasis. The World Health Organization identifies 19 fungal pathogens as either critical, high, or medium priority as public health threats. So the 4 critical priority organisms. So if we had to say what are your top 4 yeast infections that are the most dangerous, are Cryptococcus neoformans, Candida auris, Aspergillus fumigatus, and Candida albicans. And these are really worrisome yeast infections to have because we are developing antifungal resistance to our medications. The fungal cells and human cells have a shared metabolic and physiologic pathway as both are eukaryocytes, which is different than when we talked about bacteria, right, which are prokaryocytes. Most of our antifungal treatments exploit the difference between, though, the human and fungal plasma membrane composition and the composition of the fungal cell wall. Okay. So antifungal drugs are going to typically work on the differences between our plasma membrane composition and the composition of the fungal cell wall. Common drugs that you're going to see in this class are the azoles, azoles, like fluconazole, itraconazole, and then also we have allamines, elalamines, which are like the terbinafine. So azoles and the allamines block the production of ergosterol, which is the lipid component of that fungal cell membrane that is not found in humans. So we target the one thing that we know is different that is going to disrupt the fungal cell wall and help kill it off. So let's jump over to our osmosis and watch that video that I've added there. Move things over here for you. Lengthen everything out. All right.
Candida is a yeast, not the
This is a pretty in-depth video about Candida, but it's a really good example to learn more about it.
in North America. Although Candida can be found in Canada as well. Candida sometimes causes a mild yeast infection, but in some situations it can get into the bloodstream and cause severe illness. Now, there are various types of Candida species, and over 20 of them can cause disease in humans. There's C. albicans, C. parapsilosis, C. tropicalis, C. glabrata, C. cruce, C. auris, and the list goes on. Of these, the most common one is C. albicans. Candida is found throughout the body. It likes warm, moist environments like the mouth, the diaper region of babies, and in women it can be found in the vagina. Now, it's normal for microbes like bacteria, fungi, and viruses to live all over the body, but each microbe is slightly different in terms of whether it's colonizing the body, in other words, just living and not causing any problems, or whether it's infecting the body, which is causing some degree of tissue damage or destruction. An important factor is exactly how much of a microbe is present. Candida is considered an opportunistic microbe. When the amount of Candida is relatively low, it's harmless. But if a person's immune system is weakened, or if there's less competition for the Candida, then the amount of Candida can increase, and that's called Candida overgrowth. Now, Candida can exist in multiple forms. It's kind of like a chameleon. All Candida species can take on a round or oval shape, and these are called yeast cells. One Candida species, called Candida albicans, can also appear like pseudohyphae, where it looks like long, thin filaments, kind of like a segmented cactus plant. So these two shapes of Candida albicans express different protein profiles, and they give the cells different properties. In yeast mode, Candida is better at moving from one part of the body to another, whereas in filamentous mode, it's better at invading tissues. Candida typically lives on the skin or mucous membranes, and when it starts to overgrow, it can damage nearby tissue. With this, there are a few patterns of injury. The most common one is pseudomembranous candidiasis, and it's primarily due to a weakened immune system that allows for Candida overgrowth. The result is destruction of the stratified squamous epithelium layer, which is the outermost layer of the skin or mucous membranes. This causes accumulation of the destroyed cells in the keratin protein that fills the outermost layer, forming a white lesion called a pseudomembrane that looks kind of like cottage cheese. The white lesions aren't typically painful, and they can be scraped away with a tongue depressor, leaving behind a red mucosal base which sometimes bleeds. Since the underlying cause is a weakened immune system, it's fairly common in young infants and the elderly, both groups that have a relatively weak immune system. It can also be related to an immunosuppressive condition, like diabetes or HIV, or from immunosuppressive medical treatments, like steroids, including inhaled steroids, as well as radiotherapy or chemotherapy. Another pattern of injury is called erythematous candidiasis, and that typically results from a change in the levels of microbial competition keeping Candida in check. For example, a course of antibiotics or tobacco smoking, which both selectively destroy certain bacterial populations more than they affect Candida. The opposite is true as well. Sometimes there are mechanical devices like braces that favor Candida growth more than the growth of other microbes. In either situation, the result is an overgrowth of Candida, which causes increased blood flow to the affected tissue with red, painful lesions. In a lot of situations, there's a mixed pattern of injury, with both a pseudomembranous and erythematous component. Candidal infections can affect various parts of the body. The most common location is in the mouth, where it causes thrush, which is usually a pseudomembranous candidiasis. So classically, it looks like white cottage cheese on the buccal and gingival mucosa. Candida can also affect the esophagus, resulting in an esophagitis that can cause pain with swallowing. Candida can affect the diaper region of babies, causing a diaper rash with redness surrounded by scattered red spots that are called satellite lesions, because they look like tiny red satellites that are not contiguous with the rest of the rash. Another common one is vulvovaginitis, commonly called a yeast infection in women, and it can cause vaginal itching and discharge as well as pain while urinating. Now, in addition to local infections, Candida can cause more serious invasive infections, and these often develop in individuals that have underlying immunodeficiencies. Other risk factors for serious candidal infections include hyperglycemia and reduced stomach acidity. The most common situation is an infection of a prostatic device like a central venous catheter. In these settings, Candida forms a biofilm, which is where the yeast lives inside a jelly-like matrix of proteins and behaves more like a large colony than like a collection of individual cells. Candida is able to infect devices, and if the infection isn't treated, then over time, small clumps of the yeast-laden biofilm can break away, get into the blood, and cause candidemia. From the blood, Candida can spread to other locations, which is known as invasive candidiasis. Commonly affected sites are the heart valves, the liver, and spleen, as well as the bones and joints. Candida can also cause kidney infections, either as a complication of candidemia or as a complication of urinary tract infection. Candida can also cause meningitis, in particular if it infects a device in that space, like a ventricular shunt. What's more is that Candida can cause particularly worrisome infections of the eye. Diagnosis of oral candidiasis can be confirmed with a culture, and ultimately a tissue biopsy might be done in some cases as well. For invasive infections, the gold standard of diagnosis is a positive blood culture, but blood cultures can be negative a lot of the time. Alternatively, antigen testing can also be done. For example, one test looks for a cell wall component called beta-D-glucan, which is found in Candida and a few other fungal species. Another test is Candida PCR, which detects candidal DNA. Treatment of candidiasis depends on the location and severity of the infection. Oral thrush is treated with oral nystatin suspension, whereas vulvovaginitis and skin infections are treated with topical antifungals. Resistant infections are often treated with azole antifungal medications. In severe infections, especially when there's a prosthetic device like a catheter or a ventricular shunt that's infected with a biofilm, it's often necessary to remove the device. Severe infections are often treated with a medication like amphotericin, azole antifungals, or echinocandins, like micafungin. Alright, as a quick recap, Candida is an opportunistic fungus that can overgrow in situations where there are fewer competitive microorganisms or a weakened immune system. Common sites of infection include the mouth, the esophagus, the vulvovaginal region, and the groin in infants. These superficial infections are usually treated with Nystatin, or topical antifungals. Severe infections include those where a biofilm forms over a prosthetic device, and typically require removal of the infected device.
We say Neostin, right? But hey, whatever. Nice stat. Okay, so that's a great review on the review of Candida. I'm going to play a quick piece of the antifungal azoles that are listed on, or that is on osmosis. We're not going to listen to the whole entire thing, but it's a good review.
The azole antifungals are a family of medications used to treat mycoses, or fungal infections. Mycoses can be superficial, like on the skin or hair, or develop into systemic infections in immunodeficient people. Antifungals work either through fungistatic action, meaning that they inhibit fungal growth, or through fungicidal action, meaning they kill the fungi. Azoles are divided into two major families of antifungals, imidazoles and triazoles. Most fungal cells have a tough outer cell wall and an inner cell.
The eukaryotic organisms we're talking about, and fungi can be, like we talked about, yeasts or mold. So before we were talking about the yeast, the form of Candida, right? But so now we're just talking the broader fungi category.
Cell membrane. The cell membrane is mostly made of phospholipids, with some sterol or modified steroid molecules mixed in. Humans have cholesterol, while fungi have ergosterol. But both sterol molecules help keep the cell membrane stable at a wide range of temperatures. Now, the precursor to both molecules is linosterol. Fungi have a cytochrome P450 enzyme called 14-alpha demethylase in their mitochondria and endoplasmic reticulums, which converts linosterol into ergosterol. Without ergosterol, the structure of the cell membrane will be disrupted. This will cause membrane-bound proteins like ion channels to stop working properly. The membrane also becomes fragile, which eventually leads to inhibition of fungal growth. The azole antifungals include imidazoles like clotrimazole, isoconazole, miconazole, butaconazole, fenticonazole, and ketoconazole, as well as triazoles like fluconazole, itraconazole, and voriconazole. The only difference between the two groups is that imidazoles contain an imidazole ring, while triazoles contain triazole rings. These medications work by inhibiting the 14-alpha demethylase enzyme. However, they also inhibit human cytochrome P450 enzymes to a lesser degree. Because of this, the vast majority of azoles come in topical form, and they're commonly used to treat superficial mycoses. Azole creams are used for treating dermatophytosis, or ringworm, which is a red, itchy, scaly, circular rash on the feet, groin, or armpit area, caused by the trichophyton fungus. These fungi are also responsible for the more common athlete's foot or jock itch. Additionally, azoles can be used to treat tinea versicolor, caused by Malassezia globosa. This disease is characterized by hypopigmentation or hyperpigmentation in patches of skin. Last but not least, topical azoles are also used to treat yeast infections, such as oral or vaginal candidiasis, caused by the Candida species. Topical use of azole antifungals is generally considered to be safe, since absorption through the skin is minimal, and the most common side effect is skin irritation.
There, but we're going to move on from those into next the protozoa. All right, key elements of protozoa. They are also eukaryotic cells. They're abundant in our environment, and again, typically they are not pathogenic. They are these single-celled Eukaryocytes that are abundant, think soil and water. Think of things like malaria, trichomoniasis, gyaradosis, those things. Okay. The major morphological distinctions will exist between the different protozoa. So we have flagellates, amoeboids, sporozoans, and microsporidia. So we have the different kinds of these morphological forms of protozoa. When we talk about the morphologic distinctions, we're going to think about the model of movement that the protozoa use, as well as the aspects of their complex life cycles. Some protozoans reproduce by binary fission, whereas others have a form of sexual reproduction in their life cycle. Other classification schemes are organized by their site of affection, including intestinal protozoa, urogenital protozoa, and then blood and tissue protozoa. Some antibacterial agents are effective in treating certain kinds of protozoal infections. The example in the book is that you can use metronidazole to treat intestinal giardia or vaginal trichomonas infections. I'm going to go back to a YouTube video because I think it's helpful to hear about these in another fashion to kind of explain also parasites, the protozoa, and the worms and those kind of things. So let's jump in here.
In this video, we will focus on protozoan parasites. To recap, parasites can be divided into three main types: protozoa, helminths, and ectoparasites. Protozoa are single-celled eukaryotic organisms. Protozoans are a class of protists, and so are other organisms such as slime molds and algae. All these guys are unicellular eukaryotes. Protozoan comes from the Latin and Greek to mean first animal, thought to be the earliest animal to have existed. All protozoans are eukaryotes and therefore possesses what's called a true or membrane-bound nucleus. However, protozoans have no cell wall. Protozoans can either be free-living or parasitic in nature, meaning they can live in the outside world without any issues, mostly in the water, or they can be parasitic in nature and live and infect humans. Protozoa range in size from slightly more than 1 to more than 100 micrometers. They are single-celled organisms and have a true membrane-bound nucleus, but they do not have a cell wall as mentioned. Protozoa obtain nutrition from absorption of small molecules or by ingestion of them from the environment. They are predators preying on bacteria, algae, and even other protozoa species. The cytoplasm is frequently divided into the inner endoplasm and thin outer ectoplasm. The endoplasm contains granules, where they store their foods. The ectoplasm contains organelles, which are mainly responsible for movement. These organelles responsible for movement include pseudopods, cilia, or flagella. It is the mode of movement of the protozoa that is used to help classify them into four groups: the amoeba, also known as the sarcodina, the flagellates, the mastigophora, the ciliates, the ciliophora, and the sporozoa, the apicomplexa. Let's focus on each of these groups and learn a bit about their unique characteristics. Beginning with the amoeba, also known as the sarcodina. These guys live in freshwater, sea, or moist soil. The movement is by pseudopodia. They extend the cell membrane and allow the cytoplasm to flow into this extension, and thus they're able to move in this way. They capture their prey also by pseudopodia through phagocytosis, engulfing things from the outside. The reproduction is by binary fission, and they also can form cysts. An example of a pathogenic amoeba is Entamoeba histolytica. Entamoeba histolytica is a major cause of amoebic dysentery, severe diarrhea worldwide. The cysts and trophozoites are ingested in humans from fecally contaminated food, water, or hands, or from fecal contact during sexual practices. The cysts and trophozoites travel to the large intestine. Here, they can remain, causing diarrhea and colitis. They can also be pooped out, or, rarely, they can invade the bloodstream and travel to other organs such as the brain, liver, and lungs. The next group of protozoan parasites are the flagellates, the mastigophora. As the name suggests, they have flagella that help them with movement. Their body is covered by something called cuticle or pellicle. They are oval in shape, and reproduction is again by binary fission. Some examples of pathogenic flagellates include Trypanosoma, Giardia, Leishmania, and Trichomonas. Trypanosoma cruce is transmitted by triatomine bugs and causes Chagas disease, a condition characterized by heart and gastrointestinal lesions in a chronic phase of this disease. Giardia lamblia causes giardiasis. The cysts of Giardia lamblia are ingested from contaminated water, food, or fomites and travel in the small intestines, where they become trophozoites. At this stage, they attach to the small intestine and cause watery diarrhea. Leishmania, transmitted by sandflies, can cause leishmaniasis. Clinical manifestations range from self-limiting skin ulcers, known as oriental sores, to a highly lethal infection of the reticuloendothelial system, termed kala azar. Trichomonas is a very common cause of sexual transmitted infections, specifically from Trichomonas vaginalis. And there's a separate video on this. The third group of protozoan parasite are the ciliates, the ciliophora. They derive their name from the Latin word for eyelash, which describes the appearance of many ciliates quite well. Some or all the surface of the ciliate is covered with relatively short, dense, hair-like structures, the cilia, which beat to propel the ciliate through the water and or to draw in food particles. They are aquatic. They are abundant in almost every environment with liquid water, the ocean, marine sediments, lakes, ponds, rivers. Examples of a pathogenic ciliate is Balantidium coli. It is the only ciliate known to be capable of infecting humans. It is often associated with swine, the pig, as the primary reservoir host. The cysts of Balantidium coli are ingested through contaminated food and water. They become trophozoites and colonize the large intestine. They replicate through binary fission. They can cause acute or chronic abdominal pain or symptoms complicated by diarrhea or dysentery. Finally, the fourth group are the sporozoa, the apicomplexa. These guys don't have any specialized organelles for locomotion. They are non-motile protozoa. Examples include Plasmodium, which is the cause of malaria and requires the Anopheles mosquito for transmission. Toxoplasma gondii is a non-motile sporozoan that is carried by cats. It has a serious effect in immunocompromised people and can cause congenital defects. Cryptosporidium and Microsporidium causes chronic diarrhea also in immunocompromised people. So in summary, protozoan are a type of unicellular eukaryotes which are known to be one of the first living animals in the world. They are eukaryotic because they have a true membrane-bound nucleus. However, they have no cell wall. They're classified into groups based on how they move. These are four groups: the amoeba, flagellates, ciliates, and the sporozoa, which are actually non-motile. Thank you for watching.
Because we talk more about these later on down the line. Let's move on. The book goes on to pathogen identification, and this is a nice recap for us to understand the clinical concepts then in infectious disease. So when you have a suspected infection on an accessible surface, like the skin or the mucosa, oftentimes we take a swab and we send it off to lab. So what are the ways that we can do these things, and how does that work? One of the ones they talk about is a culture or an antigen identification, or we use like PCR, which is nucleic acid amplification testing. So when they talked about culture methods, they give us a nice little brief paragraph for that. It's a multi-step process. You have to have a lot of steps: inoculation, incubation, isolation, inspection, and then ultimately identification. The antigen identification, like enzyme immunoassays or the EIAs. The advantage of an antigen test are rapid turnaround and low expense, but they also have a lower specificity and sensitivity for certain organisms. So oftentimes you have to have a follow-up test to confirm. Our nucleic acid-based tests are the molecular signature tests. These are continuing to expand, and they are considered the most accurate and sensitive for pathogen identification. So one of the examples is the polymerase chain reaction, PCR, and then you'll see nucleic acid amplification test, NAAT. The NAAT are the standard diagnosis for certain infections, specifically chlamydia and gonorrhea and tuberculosis. The book talks a little bit about serology as well, as that's looking for the presence of those specific antibodies to indicate the occurrence or timeline of exposure to a pathogen. Those are things like when we test your immunoglobulins. Then the book kind of jumps right into antimicrobial resistance. So we've kind of wrapped up the different pathogens. We talked very briefly about how we test for those pathogens, and then they jump directly into these drug-resistant pathogens. So when we first got penicillin, it was life-changing. People died at a much higher rate before penicillin. However, the bacteria and microbes have responded and grown and developed resistance. Many human pathogens have rapid proliferation and high mutation rates, and that favors the development of drug resistance. Bacterial drug resistance is facilitated by small DNA elements, the plasmids, that can carry resistance genes and then can be shared by bacteria during binary fusion. So the bacteria have grown smarter, and they've changed and evolved to now form ways to get around our drugs. This we've learned the hard way about antibiotic resistance or antimicrobial resistance, and that is why now antibiotic stewardship is what it is. 20 years ago in practice, we just did not have huge antibiotic stewardship teams in every hospital. Now every facility has some kind of form of antibiotic stewardship. As a practitioner, when you're writing for antibiotics, there's somebody that is reviewing why you wrote what for one antibiotic. The timeline that's provided by your book I thought is pretty cool. It talks about penicillin in 1941, vancomycin 1958, amphotericin B 1959, the cephalosporins in the 80s, imipenem in 1985, Cipro, fluconazole, XYZ. And now we've become a lot smarter on our antibiotic stewardship because the bacteria and these bugs have gotten a lot more resistant. Genetically acquired resistance can develop from spontaneous mutations, which are promoted by the rapid cell cycle of bacterial growth and the horizontal gene transfer of those plasmids. The mechanism of the drug resistance depends on the type of the organism. Pathogens can develop or acquire the genes encoding enzymes that directly alter an antibiotic. This is kind of different, a little bit crazier. So an example would be the production of beta-lactamases by various bacteria. So beta-lactamases are enzymes that hydrolyze the beta-lactam antibiotics, such as penicillins and some of the cephalosporins, essentially rendering them ineffective. So the bacteria change something about themselves so that those antibiotics can no longer work. You can also have mutations in antibiotic target cells, such as the modification of penicillin-binding proteins, the cell wall synthesizing enzymes targeted by those beta-lactam antibiotics. I've added the osmosis link here, which is a great, again, a very good illustration of antibiotic resistance. So let's jump over back to our buddies over at the lovely osmosis.
Become highly resistant to antimicrobials.
Well, let's start at the beginning. That might be good. There we go.
The discovery of antibiotics is one of the most important advancements in clinical medicine and public health. It's laid the foundation for a number of other advancements, including the ability to perform surgeries more safely and the reduction of infant and maternal mortality rates. Many antibiotics are derived from either bacteria or fungi. For example, penicillin, which is secreted by the fungus Penicillium, can kill bacteria. This is because microbes use antibiotics to fight off other microbes. But the use of antibiotics, and more broadly antimicrobials, which include medications that target not only bacteria, but also viruses and fungi, has exploded in recent years. Antimicrobials have been used on an industrial scale, partially because of overprescription in humans, but more so because of routine use in farm animals. In fact, a good number of antimicrobials are excreted from humans and animals unchanged, and these get flushed into wastewater. which allows pathogens to be perpetually exposed to antimicrobials. In response to this enormous selective pressure, many pathogens have become highly resistant to antimicrobials. Now, when it comes to bacteria, generally speaking, there are 4 mechanisms for how they become resistant to antimicrobials. The first mechanism is antibiotic inactivation or modification, which is where bacteria develop specific enzymes that destroy and inactivate antimicrobials. One example is beta-lactamase, which is a bacterial enzyme that destroys antimicrobials that contain a beta-lactam ring, like penicillins and cephalosporins. As a result, bacteria that produce beta-lactamases are immune to the action of many beta-lactam antibiotics. The second mechanism is the alteration of a target or binding site. An antibiotic that can't bind anywhere is rendered useless. One example is a methicillin-resistant Staphylococcus aureus, or MRSA, which modifies its penicillin binding locations, making them unable to be bound by any antibiotics in the penicillin family. The third mechanism is by bypassing metabolic inhibition. For example, some antibiotics, such as sulfonamides, disturb the metabolic pathway that synthesizes folic acid, which is vital to DNA-based organisms. But to get around this, some bacteria are able to scavenge for folic acid from the environment, completely circumventing the action of sulfonamides. The fourth mechanism relies on preventing antibiotic accumulation. One way that bacteria do that is by decreasing the permeability of their membrane to the antibiotic. The other way is to create efflux pumps, which pump the antibiotic out of the bacteria. Both actions result in lower intracellular antibiotic concentrations, which reduces or even negates the effect. Now, these mechanisms work in different organisms against different antibiotics. It's an ongoing challenge. For every new antibiotic that's identified, pathogens rely on selection pressure to find a mutation that somehow allows them to thrive in the presence of that antibiotic. Alright, as a quick recap, bacteria can achieve antibiotic resistance through 4 fundamental mechanisms. Inactivation physically destroys the antibiotics before they can do their job. Alteration of the binding site leaves the antibiotic without a binding site, and therefore unable to work. Alteration of the metabolic pathway completely bypasses the activity of the antibiotic. Finally, reduction of accumulation keeps the intracellular levels of antibiotics low.
As always, a really good quick illustration from osmosis, which leads us to this slide of those different things that they already talked about there, which are also talked about in your text. All right, to review some key concepts before we move on to part 2. These are listed here. The immune system, which we're going to cover in chapter 6, is able to detect these pathogen structures and respond with both our innate and our adaptive immune system. Remember that the development of our immune response starts in the neonatal exposure and goes throughout childhood. We talked about many organisms can colonize the body without actually causing infection. And we also reviewed these key concepts as well. All right, I'm going to wrap up here, part 1, and we'll continue in part 2.